Transport across membranes. So when we're bringing materials in or past a cell membrane, there's essentially two major types, or basically we can classify them as whether they happen automatically without any energy required. That would be passive transport. They do not require any energy. ATP is a form of energy that the cells use, or whether they do require energy. Active transport, where they are actually consuming this ATP, which again is a storage of energy for cells. So let's talk about passive transport first. The first one is diffusion, and again, this is not specific to cell membranes. This will happen anywhere, where we have the movement of particles from an area of high concentration to an area of low concentration. So if you have a bunch of particles in one area, they will tend to spread out from that area. That is diffusion. And they will do this until they are about equal in both areas. They don't remain stuck in that area. So we refer to this as dynamic equilibrium. So if we had a container and all the particles were over on one side, they will spread themselves out, and eventually you will have the particles spread out evenly throughout the container. Now they don't stay stationary, but they are relatively equal. So they are called being in equilibrium, but it is known as dynamic equilibrium because one could move to one side, the other one could move to the other side. They're constantly moving, but they're equally spread out between the two spaces. So this can happen on its own or with some help. If it is something that is being helped, we call it facilitated diffusion, or it could happen on its own like what we have with osmosis. And for example, osmosis is particularly talking about the movement of water on its own, but again, still diffusion, but specifically the diffusion of water. So a couple of terms that we want to be able to discuss. Remember, solute is the stuff that is dissolved in the solvent. So normally waters are solvent in biology. Could be other things, but normally water is the solvent. And so if you have something dissolved in it, like sodium ions or glucose or something like that, the ions, the glucose, that would be the solute that is in the water solvent. With large molecules, they tend to be harder to get through the membrane itself. So if they're large, they're going to have some difficulty. Or if there's other reasons why they are not wanting to leave the aqueous environment around the cells, they can also be difficult to get through the membrane. So let's look at those types. So for example, if a molecule is hydrophobic, again, it doesn't really get that attracted to water. So if you look at these molecules here, these are all non-polar, and so they don't have a particularly easy time staying in water. You can get them to go in water, but the water's not particularly attracted to them. They're pretty easy to get through the cell membrane because remember, the inside of the cell membrane is also non-polar. And so if you have a non-polar molecule, as long as it's relatively small, it has no problem getting through the cell membrane. Also, if the molecule is small, like water, even if it is polar, as long as it's small enough, it will also be able to get through the cell membrane. And uncharged molecules that don't have a huge charge to them. So for example, glycerol, we do have those OH groups on there, but overall it's fairly symmetrical, so it doesn't have a huge overall charge to it. It's also not terribly large. It can also get through the cell membrane as well. If they are large and have a large molecule like glucose or even sucrose, they will not be able to get through. Ions with a strong charge to them also will not be able to get through the cell membrane, and again, they will not be able to pass through that non-polar hydrophobic section of the cell membrane, the fatty acid tails of the phospholipid bilayer. So facilitated diffusion, this is where you're having the particles move from high to low concentration, but it is happening with help. And so here's some of the proteins that can be involved in allowing those molecules to go through. For example, glucose is too large to go through the phospholipid bilayer, so we need glucose to travel through our cell membranes. So we're going to need a thing that will facilitate that to happen, and we use a transport protein to do that. Two types of transport proteins that we're going to look at. Channel proteins, So again, if you're dealing with something that is hydrophilic, like ions, even with water, though it can go on its own, we can have a channel protein to do that. And basically that channel protein can be open or closed, or it could be a carrier protein, which is going to be very specific to a certain thing that it's trying to bring, a certain solute that it's trying to bring through the cell membrane. So think of basically a channel protein as a doorway that can open or close. And again, whatever the particles are, they'll just move to whichever area has lower concentration. Once that door is open, they will move through in whatever way gets them to diffuse to the area with a lower concentration. Same thing is true for the carrier protein, but it works by changing its shape. So it's like these revolving door type things. It'll actually go in and it'll interact with that protein, and then it'll be able to go through. And again, this is in either direction. It'll move in whatever direction has the lower concentration. Both of these, no energy is being used. These are both passive transports. Osmosis, again, is specifically the diffusion of water, and as long as water can go through the membrane, so we refer to this as a semi-permeable membrane, the water is able to move through it. It will do so, and it'll move from the area of high concentration to low concentration. The thing that can change the concentration of water is what else is in the solution. So if you have one area that has, both of them are going to be aqueous solutions, but if one has more ions on one side, that's essentially the same thing as saying having less water. So it'll move to the side that has more ions, or whatever the thing is that happens to be dissolved. So it could be sugar molecules. Generally speaking, more solute in it would have less water, so water will move towards that side. This can create osmotic pressure, and again, this basically takes the water from an area of high concentration of water to an area of low concentration of water. So if you have high solute, that's the same thing as saying low water, right? Because it's sort of the solute's taking up all the space. So the water will move to the area that has high solute concentration through osmosis. For example, if you have a solution that has lots of solute in it, so let's say this solution here has lots of solute in it. If you had a cell in that solution, the water will move out of the cell, and it's moving to the area of high solute concentration. The name for high solute concentration, remember the prefix hyper is moving sort of high or above. So hypertonic, tonic referring to the ions in there or the solutes in there, if whatever the solutes are. So if it is hypertonic, water moves towards the actual solution it's in, which means leaving the cell. So if you have an animal cell without a cell wall, it'll essentially shrink. It'll lose all its water into the solution. The plant cell, same thing will happen, but because they have a cell wall, these solutes, the high solute concentration, the hypertonic solution, will take the water out of the plant cell, but it tends not to collapse to the same extent that animal cells do, because the cell wall maintains some rigidity, though it would be shriveled. Net movement of water out of the cells into the hypertonic solution. Remember, hyper meaning above, tonic meaning ion, so that is a hypertonic solution. Hypotonic, as you'd imagine, this means below, so there is less ions in the solution than there are in the cell. So if you imagine the cell has a bunch of solutes in it, the sugars and ions and everything else that's in there, and the solution outside just has a little bit of those solutes in it. And so in this case, the water, which is a higher concentration outside the cell, because there's lower solute concentration, will move into the cell. That's a problem for animal cells because, again, they can take on so much water that they're, as the diagram is showing here, they can burst. They essentially, their cell membrane can't hold in that much amount of stuff, and so the cell membrane will expand to the point of the cell membrane breaking apart, and the cell would die. Plants, however, because of their cell wall, they kind of like this condition. They will move water into their cells, and their cell wall will keep them from getting too full, and so essentially they will fill up with water. This is called turgor pressure, where they move water into the cell because, again, there's more solutes inside the cell than outside, and so it'll move into the plant cell, and this gives the plant cell some rigidity. The solution, again, is hypotonic, meaning that the solution that the cells are in have less solute in them, which is kind of the same thing as saying having more water in them. And so the water will move from the higher concentration into the cell where there is a lower concentration of water. Then we have isotonic solution. This is where the solute concentration inside the cell matches the solute concentration outside the cell. And it's not that water doesn't move. It is a dynamic equilibrium where you have water moving out, but for every water molecule that moves out, you have one moving in, so the net change is 0. This is good for animal cells. They want to be in an isotonic solution. Again, plants prefer having a hypotonic solution because they want to maintain that rigidity, and they do that through turgor pressure by having extra water moving into their cells. So plant cells want to be in a hypotonic environment. Animal cells would want to be in an isotonic environment. Otherwise, they've got to do some work. So iso is referring to equal solute concentration and therefore equal water concentration. There's no net movement. Still definite movement, but no net movement in or out. So with the topics that we've talked about here, again, we can get an idea of what would happen if we had a semi-permeable membrane, so something like a cell wall, but like dialysis tubing would work as well. Something that allows water to transfer through it, we can refer to as a semi-permeable membrane. So if you had a U-tube set up, this is a U-tube, with the hypotonic solution on the left and a hypertonic solution on the right, equal levels, what we'd see is that, again, there's more solute on the right than there is on the left. And so water will move towards the side. The solutes can't go through the membrane in this case here, so this is semi-permeable only to water. The solutes are stuck on one side. So the water will move to the concentration that has higher solute, or the higher solute concentration, the hypertonic solution. And what you'd end up with, again, the solutes can't move through the membrane, so the water will actually move to the one side until their concentrations are equal. All right, active transport. This requires the energy molecule that cells use called ATP, and we're going to look at it with pumps and with vesicle transport. So pumps, as their name would suggest, pump things from one side to the other, and they're moving against the concentration gradient. So this is not passive. You have to put energy in if you want things to move against a concentration gradient. They always want to, on their own, move from an area of a high concentration to low concentration. So if you want to fight that, you've got to spend some energy. So what's going to happen is these particles that you're trying to pump, say it's like, I don't know, glucose or some protein or something like that, whatever the particles are, they're going to bind to a transport protein, and that protein will use this energy molecule to sort of reconfigure its shape and move the particle across the membrane. So think of this kind of like an escalator, right? So even if the door is open, if you want to move from this area of low concentration and go to an area of high concentration, that will not happen on its own. You have to have some energy going into this, and then it can pump it to an area of high concentration, because naturally it's going to want to come back down. And if you want to fight that, you've got to spend some energy. Active transport. So for primary active transport, what happens is ions are moved in a way to make a concentration gradient. So you can imagine if we had a membrane phospholipid bilayer here, we can pump a whole bunch of ions, and this would require some energy, but let's say these are positively charged ions. We can move them all to one side of the, or a bunch of them to one side of the membrane, and you'd end up with a higher concentration. So this is the higher concentration, this is the lower concentration, and this establishes a concentration gradient. To do that, to move these to the higher side, is going to require some energy. But what you do, you end up setting up a potential difference between the two. You have more of one charge on one side than on the other, and this is called an electrochemical gradient. This gradient can then be used for secondary active transport, where we have this gradient set up and we can use it to move other things. So two ways this can work. Symport, where these positive charged ions, they're going to want to move back, right? Because through diffusion, you always have them wanting to go from an area of high concentration to low concentration. So they're going to want to do that. And with symport, we couple that with allowing another substance to move through. So let's say we had some other substance out here that we wanted to move in. because the ions are going to want to move in, we can couple that and allow the movement of these other desirable things, these orange solutes here, let's say, and they'll be coming with the ions back in down the concentration gradient. So that is referred to as symport. Antiport, you're using the same concentration gradient, but let's say we've got some ions that we want to move outside. We essentially do the same thing as symport, but it moves in the opposite direction as the ions. So as the ions come in, instead of moving something in with it, we move these other things out with it. They're moving against or counteracting each other. So for every positive ion that comes in down through the concentration gradient, we would be moving out something in the opposite direction. That's antiport. Vesicle transport is the other type that we're going to be looking at, and we've seen vesicles forming. They're sort of these packages surrounded by basically the cell membrane or something very similar to the cell membrane, but we can move large particles. So if you have these large things that don't easily fit through a channel protein, you can actually package them up in this sort of membrane-like package. That the package itself is like the cell membrane, and it can move those things in or out of the cell. And actually out of the cell, they'd be making it first. When it goes into the cell, it actually is directly using the cell membrane to do that. So large proteins, large polysaccharides, too big to fit through these channel proteins, so we're going to have to use vesicle transport. Again, using the membrane to bring these things in, the membrane essentially is going to pinch off, and then we have a vesicle inside the cell, or vice versa. So endo and exocytosis are the two major types, and as their name would suggest, endo means moving into the cell, exo means moving out of the cell. So endocytosis, again, we're going to be bringing material into the cell, and the membrane is essentially going to surround whatever it is you want to move in, pinch off, and then it'll be inside the cell as a vesicle. Three types that we're going to mention. Pinocytosis is called, referred to as cell drinking. You can think of it like that, but this is liquids moving into the cell. Phagocytosis, think about eating it. And so this is, again, large molecules or even other cells, right? Some cells will consume other cells in order to destroy them and get materials and get energy. And so this is done through phagocytosis. And then we have receptor-mediated endocytosis. So this is using particular receptors on the cell's surface to bring in, say, particular molecules into the cell. So here's a diagram of the three of them. So again, if this was some large particle that was too large to bring in through a protein, channel protein, definitely too large to fit through the membrane itself, what can happen is that the cell membrane can sort of wrap around it, and then it'll actually pinch off. So you can imagine it's sort of pinched off like that, and then it forms its own vesicle. The cell membrane reforms, and the vesicle is now inside the cell, and it can be transported to wherever it happens to be needed inside the cell. Pinocytosis, same idea, but sort of a smaller version of that in terms of what it's bringing in. But again, it starts pinching off around the thing it's trying to bring in. The vesicle pinches off, and then we have this vesicle that can then move to wherever it needs to be in the cell. Receptor-mediated endocytosis is the same process, but it's actually detecting the molecules using specific receptors on there. And then the vesicle is also going to have those molecules with the particularly identified receptors in there, and it is going to form the vesicle around it and then move it in there. So all the same principle, just a difference as to what is being moved through this endocytosis, the pinching off of the cell membrane. Realize the vesicle that gets created is really just a piece of the cell membrane that got pinched off. Exocytosis is again, similar process, just moving in the opposite direction. So this is taking things out of the cell. And the Golgi apparatus or Golgi complex, this is what makes the vesicle, because remember we're starting inside the cell, and so essentially these Golgi bodies are going to be creating these vesicles out of phospholipid bilayers, and those vesicles are going to move in the opposite direction, so they essentially meet with the cell membrane and then open up to the outside of the cell, and then the vesicle is now a part of the cell membrane. So when your pancreas is making insulin, the cells there are actually producing this hormone here, and they need to get out of the cells, and so it uses exocytosis to do that. Same thing with the digestive enzymes in your digestive system. Lots of different areas where they're being produced, but they are produced inside of a cell, and then they have to be moved outside of the cell, and this is done through exocytosis.